Okay, we want to talk about uh, the liver today. And what we've done so far is kind of talk about tissues in this class kind of independently. We talked about bones, muscle, heart, blood, kidneys, and lungs. Liver is our chance to start to talk about a tissue and then something much bigger. Kind of where the liver fits in an entire body situation. So we'll start our discussion of metabolism here as we get through the liver. So the liver is a large, spongy organ posterior to the diaphragm. So you've got the thoracic cavity, you've got the lungs, the heart. The border between that and the kind of digestive tract is the diaphragm. Now, because the ruminant stomach is so large, it tends to dominate the left side of the animal. You have the liver that kind of fills up the space up under the rib cage, underneath the diaphragm, on the right side. And if you're doing a liver biopsy, you're going to do that on the right side of the animal. The rumen cheats to the left. If you're ever doing it maroonish in color, looking at a placement somewhere in here. So fairly uptight, up under the ribs to protect it. That's probably a little too far forward, but gives you an idea that it's fairly large and fairly anterior in the animal. We mentioned or alluded to the circulation that's associated with the liver. And we're looking at the things that get absorbed from the digestive tract, the GI tract, the gut, are going to go to the liver first, then the heart, then the body. So this is a diagram where you can see this again. Duke's Physiology, a real good book if you want to get into this. So Duke's talks about the cardiovascular system. So this is kind of an overview, but we're going to zero in on how things are related to the liver and the guts and the stomach. So right side of the heart, you got the venous return here is kind of, I guess, a tan. And you got some white over here. So the oxygenated blood goes out. We got blood going into the intestines, the pancreas, the stomach, and to the liver. So this is oxygenated blood going to the liver. But you've got a secondary system here, the pancreas, liver, and spleen. They all have capillary beds which drain into our portal vein. Our portal vein then drains to the liver. So you've got oxygenated, nutrient poor blood flowing to the liver, and then you've got a parallel system, two capillary beds kind of overlapping that is oxygen poor because we've used up all our oxygen here, but nutrient rich. So our cells can take the oxygen from one capillary bed that runs in parallel and then take their nutrients from the other one. The liver gets first crack at all the things that drain from the digestive tract. And then the liver decides what to do with those. And this is where we start to go, how does this go through all the body? How things get delivered? How do those nutrients get processed by the liver and sent out to the body in a number of different directions? So, muscles, brain, all those things. So, gut, liver, heart, body. So this is the portal circulation, which is illustrated right here. So the liver is central to all major metabolic pathways. It does a range of things. The artificial liver is something that probably will not be, at least all its functions will not be developed in our lifetime. We could probably do an artificial pancreas for insulin and glucagon. That's probably doable. artificial lungs, artificial hearts, artificial kidneys. We can do those sorts of things, but because the liver does so many different things and they're complicated biochemical pathways, it's really hard to come up with an artificial liver. That would probably be um, near impossible. So we think about the liver, it's going to do all kinds of things on metabolic pathways. So we're going to have synthesis, it's going to create new compounds, it's going to degrade, it's going to break things down. We'll talk about that. Liver is our connector, intermediary. Things, reactions will start on another organ system, move on to the liver, and then the results leaving the liver will react with another part of the body. We already saw that in the laminitis. We've got the acidosis and the leaks associated with that. That goes to the liver. The liver responds to that with acids and inflammatory response. And those histamines move on to the feet. They swell up, and then we have poor hoof formation because we don't have the blood flow because all the tissue is swollen up and it just can't get in there. There's not enough pressure. When we're dealing with the liver, we're talking about hepatic. Hepatic is the formal name related to the liver. We have the hepatic circulation, we have the hepatocytes, which are liver cells. Anytime we have hepatic, we're referring to the liver. So, what does the liver do? We'll talk about protein synthesis and degradation. First up, we'll talk about the albumin that forms a number of different functions in the blood. So albumin is created by the liver, exported in. It's used to measure the health status or nutrition status. If we've got good protein nutrition, we have good or enough serum albumin. It's also a transporter and is a solute in our blood. So if we've got enough albumin, it helps with the osmotic balance. If we don't, we have less albumin, and we talked about before, we get that water belly that you'll see on starving kids. They're skeletal everywhere, but their gut, the water is collecting in the gut region because of the lack of albumin. We're not balancing out. Everything is not isoosmotic anymore. Fibrogen helps with our clots. We've got alpha and beta globulins. We've got our uh, ceruloplasmin and our transferrin that are transporting minerals. We got the protein carriers for lipid, high density lipoprotein, low density lipoprotein, very low density lipoproteins. Again, these are all created by the liver. We've got our serum enzymes, and we also, those of you that have been in the nutrition class, we know the liver makes all the non essential amino acids. It can create those 10 different amino acids and use them to build things. So we'll also mention that it rips proteins apart if we've got defective proteins floating in the plasma. If you got protein hormones, it might get broken down in the liver. If you've got these falling apart in some way, shape, or form, they'll be broken down into their individual amino acids. Those amino acids will be recycled both in the liver and out in the rest of the body. So, protein synthesis and degradation. Energy management, big thing in our dairy cows. We have to think about a glucose regulation. So we're going to take 3 carbon units. Lactate is 3 carbons. Propionate has 3 carbons. We're going to use them. Those are going to come together, and 2 3 carbon units make 1 6 carbon unit, which is glucose. We also take glucose, we burn it, generate ATP, generate energy to run the liver, or it can be taken and made into glycogen. We have liver glycogen, the storage form of chains of glucose molecules. It's a way to keep the glucoses around and not have them osmotically active. So that kind of takes them out of, they're no longer solutes in solution, they're taken out. Or we can export that glucose to the blood, and other parts of the body can use that. And we'll show that next week when we get to metabolism. Liver, metabolism, transport. Liver in a dairy cow does some of that. In you and I, we get a lot more lipid synthesis. In our birds, pigs, we'll do a lot more of that in the liver of those animals. In our dairy cattle, most of the lipid synthesis, the triglyceride synthesis, the fatty acid synthesis occurs in the adipose tissue. The liver of our dairy cows really doesn't like having fat around, doesn't like creating it. Excess fat in the liver causes problems, and we'll talk about that on the other side of metabolism. We also, with energy, we're thinking about cholesterol synthesis and export in our liver. In a human, we're talking about 5 grams of cholesterol produced each day for all the many functions that cholesterol does. The other function, besides creating proteins, managing energy, we talk about detoxification. Liver will take two ammonias, they'll stick them together with a carbon unit, create urea that detoxifies the ammonia, which is much more nasty than the urea. Urea will eventually go to the kidney and be removed, but in the dairy cow, it can also go up into the saliva and be recycled in the rumen. The microbes will use that to build new protein. So that's a real plus there, but we're detoxifying ammonia in the liver. We will take apart steroids as they are moved through the body, fall apart. We will break down the steroids in the liver. That's why people who do excess steroid hormones usually have liver problems. The liver is working too hard to break things down. Xenobiotic metabolism, things that are foreign to the body. The liver is one of the ways to destroy those things, to break them down, to metabolize them, to get them out of circulation, either by burning them to CO2 and water or taking them apart into their component parts. So xeno means outside, biotic, biology, organic. So alcohol, drugs, antibiotics, a lot of those are broken down in the liver. Liver trying to protect the body from things that don't belong, destroying those.